Hello, my name is Dr. Lopez.
And I'm Dr. Kim.
And today we will be explaining the DNA replication process. So the first thing that happens when replicating DNA is a DNA helicase will come and unzip the two strands of DNA, like this. Zip, zip. So today, well, for right now, I'll be focusing on the leading strand. So we'll take the lagging strand out. The reason this is the leading strand is because, as you can see, it starts in a 5 to 3 direction. And when you replicate, you have to replicate in an anti-parallel direction. And in fact, the two strands of DNA are in an anti-parallel. So when you're replicating, it always has to be in a 3 to 5 direction, which means we'll be going this way. So the first thing that happens after the DNA helicase unzips it is these single strand binding proteins will come and hold on to this side of DNA so that it's stable and it doesn't snap back together with the other side. Next, a DNA primase will come in to start the replication. After the primase is placed, the DNA polymerase will come in and it will place these nucleoside triphosphates like such. So after these are placed, the two phosphates on each nucleoside triphosphate will break off, which will result in a release of energy. So the energy that is released helps bind everything together, which then will result in another DNA side, and you get a whole new piece of DNA from the leading strand. So now we finished describing the leading strand, so we'll go into what happens in the lagging strand process.
Just like the leading strand, the single strand binding proteins will attach to this lagging strand. And the reason why this is called the lagging strand is because this is anti-parallel to the other leading strand. Therefore, we would still have to replicate in a 3 to 5 direction, so we have to replicate it in fragments called Okazaki fragments. So first, RNA primase will begin the process of replication, and DNA polymerase 3 will attach pieces of RNA to it, and then DNA polymerase will also attach pieces of DNA, thus the name Okazaki fragments. Then DNA polymerase 1 will come in and replace these chunks of RNA with chunks of DNA. So the next step then is to bind these DNA fragments and make one continuous strand.
And we do that by using DNA ligase.
Thus, the result is 2 different replicated DNA strands.
Thank you so much for watching.